Hey everyone. Right, we'll jump right into it because thanks that everyone got the memo that today's lesson was going to have a changed timing. As usual, I'm going to keep an eye out on the chat. This is quite small group teaching, so hopefully you'll be willing to contribute a little bit more, everyone. And I'm hoping you'll be able to ask more questions today, and just everyone will be more kind of willing to have like a bit of a chat about it because there really isn't that many people. So this is very, very small group teaching. So the first thing we're going to talk about is we're going to start talking about surface area to volume ratio. So this is basically the underpinning of everything else that we're going to talk about today. So everything else we're going to talk about gas exchange and diseases and really cool biology, but you need to underpin it with some knowledge and some theory. And the theory behind this is surface area to volume ratio. So remember, most exchanges in a cell happen at the exchange surfaces, which are the bits that are in contact with the external environment, right? So it's unlikely that we're going to have gas exchange kind of happening here in the middle of the cell because, again, there's really not, it's not going anywhere. It only happens at the bits that's in contact with the with outside the cell, right? So the surface area represents that total area of the parts of the organism that are in direct contact with the environment. So it's basically this area that is going to be the kind of the outside area of this environment. So in gas exchange, the cell surface area is basically representing here the total area of the cell membrane. If we had so another experiment that you would normally do is the beetroot in the cube experiment, which I'm sure all of you have done previously. This is a terrible drawing of this cube, but you'll have done so remember we use this to assume a cell membrane. So as you can see, the cell membrane, the bit on the outside is whatever is in contact with the external environment here. Cell volume is all the contents within the cell. So that's everything that's going to be in the cell. So it's going to include the bits, the membrane bits, but it's just everything that's all of the contents within the cell. So for example, to calculate the volume of a sphere, we'll do this equation, 4/3 pi r cubed. Now this is an equation that's given to you in most specifications. So you don't need to memorize it, but this is an important equation where r is the radius, pi is pi, and 4/3 is 4/3. The surface area to volume ratio will determine the rate of exchange. So the greater the surface area to volume ratio, the more efficient exchange is happening. So very small surface area and a very small volume. Large surface area has a very large volume, but really it's all about proportions. So large surface area to large volume ratio, small surface area to volume ratio. Remember, cells can divide into two new cells. These cells will each have a greater surface area to volume ratio than the one original cell. As you can see from this kind of square diagram. Small organisms generally are able to use just diffusion alone to allow for gas exchange due to their large surface area to volume ratio. Because of their, small diffusion distance, and there's also a relatively small demand for substances. Large organisms, however, cannot rely on simple diffusion. They have a low surface area to volume ratio and a huge diffusion distance. Plus, usually they generally have a higher demand for substances because they're generally more metabolically active. If you think about the more complex processes that human cells need to do relative to animal cells, then you'll realize that for context is quite an important kind of point. Large organisms have therefore they have to have specialized exchange systems. And we have organ systems that have high surface areas, and all of these organ systems then work together to maximize the efficiency of exchange. So alveoli in humans really particularly help with this. And we're going to discuss human gas exchange in a lot more detail in the next 5-10 minutes. Right, any questions so far about surface area to volume ratio? How about we give you an opportunity to do some calculations? So now have a go at this one. So try to work out the surface area to volume ratios of these ones. Oh, you'll need to measure with centimeters. Okay, you can't do that. All right, that's fine. I'll find some other questions you might be able to have a go at. Nope, same question. Okay, have a go at this one. Actually, yeah, don't worry about these. Sorry. It's fine. We'll just move on to gas exchange. Right. So there's loads and loads of ILOs that you want to be learning from here. So we're going to have a lot about gas exchange across bodies, tracheal system, fish, then leaves. So adaptations of dicotyledonous leaves, then xerophytic and hydrophytic plants, terrestrial insects, human gas exchange, alveoli epithelium, and then loads and loads of lung disease related stuff. So there's a lot to this, which means that it's going to be it's going to be quite difficult. And so let me know if I'm going too fast for you. So how does gas exchange work in basic unicellular organisms? Well, we discussed this earlier. Gas exchange works only via the cell membrane. Oxygen and carbon dioxide small enough to pass through, and they don't need any complex. They basically get all their nutrients through diffusion alone. And this is done through diffusion gradients, which we know about. Amoeba proteus is a single-celled organism which uses this system for gas exchange, for example. So an amoeba is another example of one that uses this system. Okay, tracheal system of an insect. So remember, insects usually have quite tough exoskeletons. Now this is quite thick. So if you imagine, and insects don't tend to have mouths in the classic way that humans do, where they can breathe through. So how does oxygen get in? Well, they've got basically holes in their exoskeleton called spiracles, which allow air to enter the body of the insects. And spiracles can be opened or closed and are controlled by specific specialized muscles. What are spiracles quite similar to in other organisms? In another organism, stomata, yeah. So spiracles are quite similar to stomata because they're small holes which are within that thick, waxy cuticle that plants have. Spiracles then transfer the air through thin tubes called the tracheae, and trachea are lined with chitin to prevent their collapse. What is the chitin quite similar to? Cartilage, yeah. Cartilage lining human trachea. Perfect. And then the tracheal system at the end has got a very specialized fluid, which basically carries dissolved oxygen into the cells, and CO2 then enters the fluid. The CO2 goes back up the trachea and is back out through the spiracles. Okay, any questions about insects? Again, there's only like 9 of you, so if you got any questions, just shout out or put it in the chat. Wait a second. Do you want me to go back to let you take notes on that, or is everyone okay with that? Cool. We'll move on then. So the next bit is about fish. How does gas exchange work in fish? So remember, fish live in water, so they have to extract oxygen that is dissolved in water. So to do this, they've developed a very specialized countercurrent respiratory system involving gills. So remember, because fish are in a low oxygen environment, they need to extract as much oxygen as they physically can. So they have basically, they have these gill slits, which are positioned on the side of their heads. As you can see, they'll be like zoomed in here. I don't know how many of you have seen like a fish head, but yeah. And gill slits are separated from each other by a thin, really thin piece of bone called the gill arch. So, oh, that's not really the process. Let's go into a little bit more depth on that, just so all of you guys can get a bit more understanding on that. So give me a sec. Okay, we'll go into a little bit more depth. So basically, what is going on here? So this is what is described as a countercurrent exchange system. Does anyone know of any other places where countercurrent exchange systems occur? Dialysis, yeah, dialysis is a perfect example of one where there's countercurrent exchange. But really, it happens in the loop of Henle generally in the body. That's one of the best places for countercurrent exchange. Okay, so now give me a sec. Okay. So fish ventilation. So what happens is the fish head is basically made up of show you the fish head labeled. This might be a bit terrifying for some people, but it is made up of this. It's made up of a structure. No, not those. Those are the bones. Don't really need that much detail on the fish head. Basically just looking for the yeah. So they've got basically at the bottom of their mouth, they've got something called the buccal cavity. And so what happens is when the mouth of the fish opens, that buccal cavity basically moves down, right? So therefore, what happens in the buccal cavity, the volume of that buccal cavity is going to be increased. And therefore, what's going to happen to the pressure in the buccal cavity? It's going to be reduced, right? So if volume increases, pressure is reduced. And so therefore, water is able to move into the buccal cavity down that pressure gradient. Remember that here in this part, which is protecting the gill arch, is something called the operculum. It's something called the opercular valve, right? And the opercular valve remains closed. Now, once the opercular valve or the operculum, opercular cavity has basically expanded. So the opercular cavity, the floor again moves down, increasing the volume, reducing the pressure. What the buccal cavity wants to do is it wants to increase the pressure in the buccal cavity, so water flows from the buccal cavity into the apocular cavity. And how does it do that? Well, the floor of the buccal cavity basically moves up, lowering the volume, increasing the pressure. So water then moves over through the buccal valve and through the apocular valve and into the apocular cavity. Now, when it flows into the apocular cavity, it basically is then pushed out and it then flows over the gills, allowing gas exchange to occur. Does that make sense? If you guys want a written version of that process, I'm going to put it in the, I'm going to just give you a whiteboard and I'll just write it out for you, just so you guys have that kind of the written version of how that process works. If you have any questions, let me know. And then we'll start doing plants. Okay, I'll give you like 2 minutes just to write that down. What's the apocular cavity? So the apocular cavity is the cavity just before the gills, basically. So if you want to kind of think about it from a human perspective, the buccal cavity is the mouth. The apocular cavity is kind of the pharynx, if that makes sense. It's kind of the first bit of the, just before you get into the larynx. It's that first bit in the airway. Does that make sense? What are some common exam questions that they might come up on fish respiration? Usually they'll ask maybe they'll ask you to relate fish ventilation. This isn't respiration. Respiration is the process of forming energy, ATP, from glucose. But from fish ventilation, they usually ask about basically how to relate that to other types of ventilation. So they'll ask you about kind of the similarities between fish ventilation and maybe dialysis or another complex countercurrent system process. Or they'll ask about the importance of valves, or they'll just ask you for the process and they'll give you a 5 marker on describe that entire principle to it, which I'm sure some of you will, some OCRers will know has happened a couple of times. Is this all you need to know about? Yes. You don't need to know that much depth about fish ventilation for OCRA. Did you? Yeah, I did. Yeah. I was 3rd year of the new spec. So actually, yeah. Quietly on with it. Give you another minute. Then we'll move on back to the other. Do you not need to write about the countercurrent system? You do, but I've talked about that. Basically, all you need to know is that oxygen, so the blood flows in one direction and the water flows in the opposite direction, and that maintains a constant gradient for diffusion to occur the whole way through. Oh, a graph. Yeah. Okay. Give me a sec. We'll just get on to the exchange then. Basically, so normal exchange works. This is kind of what it looks like. So blood flows in one direction, water flows in the opposite direction, and there's a maintained concentration gradient the whole way through. Whereas normal exchange kind of looks like that. Co-current, you end up having huge diffusion at one end, but counter-current, there's constant diffusion at both ends. Does that make sense? Okay, leaves of dicot plants. So remember, there's the mesophyll cells on the surface of plant leaves. There's a lower and upper epidermis. The gas exchange occurs mainly through stomata that are present mostly on the lower epidermis. So stomata are openings which allow for the exchange of CO2 and O2. And the opening and closing of stomata are controlled by guard cells. So stomata close when the plant has lost too much water, and they open when gas exchange needs to be maximized. In plant leaves, approximately 50% of the volume of a plant is airspace. Airspace are necessary to allow for circulation of oxygen, carbon dioxide, and water vapor. Plant cells are usually reliant on both O2 and on CO2. So when does gas exchange take place in plants? So again, during photosynthesis, there is gas exchange. Plants take in carbon dioxide through the stoma and release oxygen into the environment. During respiration, which occurs mostly at night, plants take in oxygen through the stoma and release carbon dioxide into their environment. So very slight differences on that. And then remember, both carbon dioxide and oxygen are exiting and moving through the stoma by simple diffusion down their concentration gradients. Doesn't respiration need to happen? Yes, but it's how relative. So basically there is a graph where respiration is happening all the time, and so is photosynthesis technically because of stored products, but respiration mainly occurs more than photosynthesis at night, and photosynthesis occurs more than respiration during the day. Does that make sense? Yeah. So it's about relative amounts. Okay. So structural adaptations. How does water loss happen in plants? So remember, plants store water in their leaves as water vapor, and water can be lost in a plant through transpiration. So during transpiration, water vapor can diffuse out of the plant leaves through the stoma. Keeping the stomata open all the time would lead to way too much water loss though. But closing stomata permanently would disrupt gas exchange. So there's a really cool system where basically you have, does anyone know the system about potassium ions and UV light and guard cell activation? Ish? Okay, so I'll explain this. Basically, what happens is when UV light is present, there is basically active transport of potassium ions into the guard cells. That causes the guard cells to basically, that causes water to, remember, water is going to go down its osmotic water potential gradient. So water goes down its water potential gradient into the guard cells, which causes the guard cells to swell. And when the guard cells swell, the stomata shut. So, and that makes sense because when there is sunlight, there's going to be a constant water vapor gradient, right? Which then means that water is going to be lost a lot. So therefore, that UV light mechanism is a brilliant way of the plant protecting itself, basically. Yeah, it does involve ABBA. You don't really need to know that much in too much detail. I think a lot of you A2 guys might, because that's to do with auxins and all of the plant hormone stuff. But for AS level, for the AS level that we're currently going through, you don't need to know it in that much detail. So the upper pole of the upper layer of the stomach of the plant leaves is waxy cuticle, which basically helps minimize water loss through transpiration. Guard cells are open during the stomata open during the day to maximize gas exchange at the same time as photosynthesis, but they can close really quickly if water is in shortage. So as you can see, water flows in, water will flow out when the guard cells are, when the water is flowing in, stomata open. When the water flows out, stomata close. And that's due to potassium ions, basically. So stomata open in the presence of UV light. It's the other way around. So in the presence of a good supply of water, the guard cells swell up and become turgid. As their volume increases, they end up pushing against each other, leading to the opening of the stoma. And turgidity of guard cells is caused by the pumping of potassium ions out of the guard cells via active transport. Pumping of potassium ions is activated by other factors, such as the presence of light and if the plant is photosynthesizing. In the absence of light, pumping of potassium ions stops. Guard cells lose their water supply, which causes them to relax and for the stoma to close. Okay, I'm just going to let you guys write that down. So that's steps 1 to 3. Give you a minute. So it's the other way around to what I was saying earlier, which is that they close when they're cool. And then steps 4 and 5. Any questions at this point? Xerophytes are basically plants that are adapted to survive in hot, dry environments. An example of this is cacti. Is there a word that you can use instead of losing turgidity? Well, becoming flaccid is the same as losing turgidity, but that would be kind of the opposite of it. Yeah. And examples of xerophytes are cacti. So their job, because they live in such a lack of an environment with a lack of water, there is a really high risk of losing water through transpiration. But they don't want this. They need to minimize this because it's going to be hard for them to retain and get that water back. So they do everything they can to not. So the first thing they do is they use a process of pitted stomata, which is where their stomata fall inwards into sunken pits. And on top of that, you can trap a layer of moist air. The water vapor in this moist air reduces that gradient between the mesophyll and the surface of the leaf, reducing water loss. They also have fewer stomata. Fewer stomata also means less water loss. A thick, waxy cuticle, again, reduces water loss through transpiration. shiny cuticle, which helps reflect much of the sunlight away so that it doesn't end up getting absorbed. Thick stems. So water stored in these thick stems minimizes water loss. And as well as large and deep root systems, which allows the amount of water that they can pick up away from the ground. They can also have pointed and sharp leaves, which minimizes the surface area for water loss. But also, if they have curled leaves, it helps trap a layer of humid insulating air. So let's go back and just take some notes down on those ones. So just write these down. I'll give you a minute for each slide. Okay, next one. And next one. How does the light cause pumping of potassium ions? So it's to do with, it's quite a complex process that you don't really need to be aware of. But it basically, it's to do with the UV light and energy from the UV light then being used as part of the pumping process. But you don't really need to know that as part of your spec. Cool. Again, water loss in insects is through spiracles. Can we think about, okay, let's think about some potential adaptations. Just bounce them off what you're thinking about how we reduce water loss in insects based on what you've just learned. So give me some ideas. Closing the spiracles, yeah. Sunken spiracles, potentially. Reducing movement. Why do you think that would help? Less movement, less metabolic demand for oxygen, potentially, but that's not going to reduce water loss per se. But let's think. So they've also got a thick exoskeleton, right? And that prevents water loss. They've got hairs on their spiracles, which help trap that layer of moist air. So similar to the way that sunken stomata work, the insects use hair to do that. Okay, so that's the first part of this. So that's all the other organisms that you need to know. The next part is quite tough. It's all the human stuff. So let's take a rain check. Any questions so far about all the other organisms that you need to know about? What about the effect of in changing the surface area? Yeah, so tracheal fluid and lactic acid changes the surface area because what it does is it changes that diffusion ability. So remember that if you have lactic acid buildup, then the demand for oxygen is greater, which therefore it means that you basically you're basically drawing in more oxygen, and so therefore it just increases that ability for diffusion to occur. Any other questions? Otherwise, we can move on to part B. Cool. So in this part, we're going to be talking about the structure of the human gas exchange system, as well as the essential features of alveoli epithelium. So how does gas exchange work in humans? Well, gas exchange in the humans is carried out by the respiratory system, as you know. So nose, nasopharynx, mouth goes into the oropharynx, which then all feeds into the laryngopharynx, Laryngopharynx then feeds into the trachea. Trachea feed into the bronchi. Bronchi feed into bronchioles. Bronchioles feed into the alveoli. So, and then that's where alveoli is where specific gas exchange occurs. And I'm sure we all know this. I'm sure we all know this labeled diagram as well. So air enters the lungs through the mouth and nose into the larynx. Trachea, air passes down the trachea. The trachea split into two bronchi. Each bronchi splits into loads of bronchioles. And then at the end of loads of bronchioles, there are loads and loads and loads of small alveoli, which are specifically specialized for gas exchange. So I'm sure you know this. So then it passes through the naso, oro, and then laryngopharynx. But remember, at this point, what happens is the air is humidified. So we basically, because normal air can be quite dry. So to overcome this, air is humidified in the pharyngeal passages. And this is really, really important because what it does is it basically allows for more efficient gas exchange to occur once the air gets into the lungs, because it reduces that it increases that diffusion gradient. The nasal passage is lined with ciliated cells, with ciliated goblet cells as well, to filter out any dust, bacteria, and viruses from inspired air. There are also mucous membranes in the nasal and oral pharynx to help humidification of the inspired air and to help trap dust and bacteria. Then the trachea, cartilaginous tube, which provides a pathway for air entering and leaving the lungs. There are tracheal cartilage along this tube, which prevents the trachea from collapsing. Goblet cells to release mucus. Ciliated cells to help trap dust and pathogens. Then the bronchi have goblets and ciliated cells to filter out dust and pathogens. The bronchi are supported by cartilaginous rings, which prevent it from collapsing. And then the bronchi splits into bronchioles, which are small, fine extensions of the bronchi, which branch into different regions of the lungs, which creates what is called the bronchial tree, because it looks like a tree with its central kind of branches and then the leaves coming off the end. They also have ciliated cells to filter the air of dust and pathogens, but they also have smooth muscle cells. And these smooth muscle cells help with bronchodilation and bronchoconstriction, as I'm sure you know that. Yeah. And the bronchioles are also, they help regulate the diameter. The bronchioles can dilate or can constrict in response to various stimuli from the nervous cells. So bronchodilation is the widening of the diameter, and it's caused by the hormone adrenaline, which helps increase airflow. Bronchoconstriction, on the other hand, helps narrow the diameter of the bronchioles, and this is caused by the hormone acetylcholine, which is mainly released to prevent further allergies and infections or excessive stress. These both occur subconsciously. They don't occur, there's no conscious regulation of this. And alveoli are the primary surface for gas exchange in most humans and vertebrate. Alveoli are also they're like tiny sacs at the end where basically they inflate. They're kind of like balloons, and the inflation helps increase their surface area. They have elastic fibers to allow for stretch and recoil, which allows them to spring back into shape after air leaves. They also produce a small fluid called pulmonary surfactant. And pulmonary surfactant is basically, it helps the alveoli keep their shape and prevents alveoli collapse. And it also helps in the removal of any trapped fluids and keeps the alveoli dry. The alveoli wall is approximately one cell thick, and the thin wall is necessary for the diffusion of gases. Alveoli are wrapped in capillaries, and this wrapping in capillaries carries deoxygenated, basically it carries deoxygenated blood from the heart to the alveoli, where CO2 diffuses out of the blood, and O2 from the alveoli diffuses into the blood. High surface area, which increases the rate and efficiency of diffusion. Right. We're gonna, because a lot of this next bit is about kind of stuff that you probably should already know, which is the simple processes of this. So deoxygenated blood from the heart travels to the lungs, CO2 out of the blood into the alveoli, O2 into the blood, newly oxygenated blood, then back into the heart. Yeah. Okay. The next bit is the mechanism of breathing. So the last thing I want to do is I want you to all list one specialization of alveoli which helps them perform their function. Thin wall. Why? One cell thick, short diffusion distance. Perfect. Covered in capillaries to create a steep diffusion gradient, but it also helps shorten the diffusion distance. Anything else? Nope. Okay, cool. We'll move on. Those were pretty much all the other ones. You could talk about a surfactant. Surfactant helps prevent it from collapsing. But yeah, either way, all of those would be fine. Okay, mechanism, biomechanics of breathing. So ventilation is basically the movement of air into and out of the thoracic cavity. The thorax or chest is made up of a rib cage which houses the lungs and the heart. Basically, during contraction, the thoracic cavity will expand and then it will contract. The expansion and contraction allows for changes in air pressure for breathing. The biomechanics of breathing, expansion and contraction is regulated by two major sets of muscles, specifically the diaphragm, as well as the intercostal muscles. So inhalation and exhalation. So when inhaled, the diaphragm and the intercostal muscles contract. So the chest wall basically moves upwards and outwards, expanding the thoracic cavity. This increase in thoracic cavity increases thoracic volume, which decreases thoracic pressure. And so therefore, air is drawn into the lungs down that pressure gradient via the trachea. In exhalation, both muscles relax. So the chest cavity basically relaxes, so everything moves back to its normal place, which then causes the air pressure to increase. The decrease in volume increases the pressure above atmospheric pressure, so then air moves out of the lungs down its pressure gradient. Does that make sense? So moving down, then moving back up. Okay, we're going to end at that. The next bit was about spirometry and kind of measuring gas exchange. If you have any questions about that, just let me know, but otherwise, we're going to kind of stop there. No worries. See you guys. Bye.